Before the mRNA that was formed in transcription can go to a ribosome and be translated into a protein, it first has to be modified. We call this post-transcriptional modification, or after transcription modification. And it's also known as alternate splicing. Alternate means that there are different variations possible, and splicing is to cut something apart and put it back together again. So the pre-mRNA that was formed in transcription actually contains a number of different segments. The ones that are labeled A, B, C, D, E, F, G, H are known as exons, and the ones that are colored in red are known as introns. Now, exons get their name because these are the segments that are eventually going to be expressed as proteins. These are the ones that are going to go to the ribosome. The red segments, or the introns, are cut out and removed. These are called introns because they are the in-between segments, and they do not lead to a protein. We don't know exactly what these introns do. It used to be thought that they were junk DNA. Currently, scientists are working on other functions that the introns may serve, but we don't know yet what those are. Now, the splicing enzymes, after they cut in between each of the introns and exons, reassemble the exons in one of any number of possibilities. So, for instance, the splicing enzymes in one type of cell might put together the exons A, C, D, F, G, and that would lead us to eventually a certain protein. We'll call it protein number 1. In another type of cell, the exons B, D, E, F, H might be used, and that would lead to a slightly different protein. We'll call it protein number 2. And in yet another type of cell, the exons may be assembled to form A, B, C, E, F, G, and that would lead to yet a different but related protein, which we'll call number 3. Therefore, one gene in DNA and one set of pre-mRNA can actually lead to a number of different genes. This helps to solve one of the mysteries that's plagued molecular biology for many years, where scientists originally thought that it was always one gene led to one protein, but we had so many more proteins in our body than we had genes that it just didn't make sense. Now scientists know that there's one gene, but it can lead to many different proteins. And this is important because every cell in your body has the same DNA in its nucleus, but not every cell has the same need for proteins. So this allows each different cell type to make the version of the protein that that cell type needs to function.